In 1928, Alexander Fleming left a petri dish uncovered in his lab. When he returned, a mold had contaminated it, and everywhere it touched, the bacteria were dead. That discovery became penicillin. For decades, antibiotics changed everything. Infections that killed millions for centuries became treatable overnight. We called it a miracle. But we forgot one thing. Bacteria had been fighting chemical warfare for billions of years before we arrived, and they were very good at it. Most antibiotics target something unique to bacteria. Some attack the cell wall, the rigid structure holding the cell together. Without it, the bacterium swells and dies. Others block the ribosome, the machine bacteria use to build proteins. Without proteins, the cell cannot survive. These targets exist in bacteria, but not in human cells. That is what makes antibiotics precision weapons. But bacteria found ways around every single one. Bacteria divide every 20 minutes. In 8 hours, one bacterium becomes 16 million. With every replication, there is a chance of mutation. Most mutations are harmless, but occasionally one gives an advantage. A modified cell wall the antibiotic cannot penetrate. A ribosome the drug cannot bind. An enzyme that breaks the antibiotic before it can act. In a population of millions, even one resistant bacterium survives. That one survivor reproduces. Within days, the entire infection is resistant. But bacteria go further. Through horizontal gene transfer, a resistant bacterium passes its resistance gene directly to a neighboring cell through physical contact, even to a completely different species. Resistance spreads not just through offspring, but through sharing. A gene that evolved in one species can be inside a completely different bacterium within hours. This is why resistance spreads faster than anyone predicted. Inside the human body, the consequences are already visible. A patient arrives with a severe infection. A first antibiotic is prescribed. It does not work. A second is tried. No response. A third, a last resort drug, fails too. Pan-resistant infections? Resistant to every available antibiotic, already exist in hospitals worldwide. Without working antibiotics, routine surgeries become dangerous. Cancer treatment becomes too risky. Minor infections turn fatal. The post-antibiotic era is not a distant warning. It is beginning now. How did we accelerate this so fast? Antibiotics prescribed for viral infections they cannot touch. The common cold, the flu. Applying pressure to bacteria with no benefit. 70% of antibiotics used globally go to livestock. Not to treat disease, but to promote growth. Patients stopping early leave the most resistant bacteria alive to multiply. We taught bacteria how to survive them. Science is fighting back. Phage therapy uses viruses that hunt and kill bacteria through mechanisms resistance cannot easily overcome. Antimicrobial peptides punch holes through bacterial membranes. CRISPR targets and destroys resistance genes directly. New antibiotic classes, the first in decades, are entering trials. The pipeline is thin. But it exists. Antibiotic resistance is here. In hospitals, in farms, in soil and rivers, carrying resistant bacteria worldwide. Every unnecessary prescription gives bacteria another chance to adapt. Every incomplete course selects for the survivors. Understanding the biology, how resistance evolves, spreads, and what it truly costs, is the first step toward reversing a crisis that is still, just barely, within our power to control. If you made it this far, you're a legend. Hit that like button, drop a subscribe so you don't miss the next one, and I'll see you guys in the next video.